Good morning, and welcome to the third annual Children's National NIAID Symposium. I'm Lisa Guay-Woodford, Director of the Clinical and Translational Science Institute at Children's National, a CTSA-funded partnership between the Children's National Hospital and the George Washington University. This symposium celebrates the programmatic partnership between Children's National and NIAID that is organized as an administrative supplement to the CTSI-CN. Given the scourge of the current global pandemic, this year's symposium will focus on COVID-19, the infectious disease caused by the SARS-CoV-2 virus. We have an outstanding slate of speakers who will take us through discussions about fundamental immunobiology, to epidemiology and clinical disease, to considerations about emerging treatment strategies. Our symposium will close with a thought-provoking presentation by Dr. Zeke Emanuel about the societal and economic impact of SARS-CoV-2. I'm delighted to report that we have over 1,100 registrants for this symposium. In the symposium, each session will be moderated by a senior leader from either Children's National or NIAID, and the presentations will be followed by a panel discussion for questions and answers. The sessions will be separated either by a 10-minute break or lunch. So with this background, let me welcome you to what promises to be an exciting day and open our symposium by introducing the senior leadership from Children's National and the NIH. Our first speaker is Dr. Kurt Newman. Dr. Newman has served as President and Chief Executive Officer of Children's National since 2011. He's a pediatric surgeon by training and a nationally recognized leader in pediatric health. Dr. Newman joined Children's National as a surgical fellow in 1984. He rose through the ranks to become Surgeon-in-Chief and Senior Vice President for the Roberts Center for Surgical Care in 2003. In that role, he was instrumental in developing the vision for the Sheikh Zayed Institute for Pediatric Surgical Innovation, which was created in 2009 through a transformational $150 million gift from the UAE. In 2017, Dr. Newman's medical memoir, Healing Children, A Surgeon's Stories from the Frontiers of Pediatric Medicine, debuted as an Amazon bestseller and earned national attention and critical praise in the New York Times Book Review, the Washington Post, and the Harvard Business Review. Dr. Newman, I'd like to invite you to give your welcoming comments.
And good morning, everyone. And thank you, Dr. Guay-Woodford. It's just a pleasure to be here this morning. It's quite an honor. And I appreciate everyone joining us here for the third annual Children's National NIAID Symposium. It's just an incredible lineup, and to use a baseball analogy, it's an all-star lineup. And of course, we have one of the world's greatest scientists and a friend of mine who's on the forefront of everyone's mind as our cleanup hitter, and that's Dr. Tony Fauci. If I could have the next slide, please. I'd like to highlight the partnership that I think is just so important and unique. Children's National, we're here in Washington, D.C., and we're ranked number 7 in the country for NIH funding for children's hospitals. But beyond that, we have multiple clinical and research partnerships across the NIH with many of the institutes. And I think it's because we share a set of values, we share a vision about doing what's right for children, and it's just so wonderful to see how we've been able to break down the silos that relate sometimes to intramural research and extramural health to bring these organizations together and interact on very pressing issues important to our nation's health. And I think the topic today, for example, is just one example of how we're doing this. Next slide. So this, in surgery, we talk about the all-star operation where all the greats came together. This is one of those photographs that I think was historic because it was the inauguration of this partnership between the Clinical and Translational Science Institute at Children's National. And Lisa, Dr. Guay-Woodford, thank you for your leadership on that. And with the NIAID and Dr. Fauci, Dr. Lane, and many others that put this together as well with Dr. Batshaw and some of my colleagues. I just think it signifies how we can take clinical research and a partnership and devoted to protecting the health of children with really incredibly complex issues and diseases through collaborative research and education. And this strategic and novel alliance is supported through the Clinical Translational Science Institute at Children's National. And this CTSI helps us to categorize many new and powerful interactions. Just to pivot a bit, I wanted to give a few of my observations around, and if I could have the next slide, around the COVID-19 response. Children's National here, we saw early on one of the highest numbers of COVID-19 patients for a children's hospital. We launched, in response to that, launched the first pediatric COVID-19 drive-through testing site in the country, having seen the value of that in countries like Korea. We thought that there needed to be something like that specific for children to help support our community. By participating in a lot of conference calls from around the world and with our colleagues at the NIH, we were one of the earliest identifiers of the MIS-C syndrome in children here in the United States. And our partnership with the NIH allowed us to move quickly into first-rate conversations about the pandemic and to jumpstart projects of importance to both organizations. And I think that will continue on, and we'll hear much more about that through the day today. Can I have the next slide, please? This drive-through site was a response to the community need. So this was based at Trinity University, and it was not for the kids that were sick that were coming to the hospital. This was for kids that might have some symptoms or have other issues or lived with people where there was a concern, and we stood this up within 6 days of deciding to do it in March. Over the course of the next few months, we saw more than 2,400 children that were tested. And I think there were many important observations from this early this early initiative. We saw that 29% of the kids, again, these were not very symptomatic, were testing positive. That was way out of proportion to what was being reported. And we also tested kids from across DC, Maryland, and Virginia, and it was a huge crowd pleaser for our community pediatricians that were using this resource. Next slide. But being an academic institution, we also analyzed these results with multiple publications, and this is, I think, one of the most important ones from Monica Goyal and our Chief Medical Officer, Denise Corr-Bramble, about the racial and ethnic and socioeconomic disparities that we were seeing. And this has prompted a lot of research and additional research and investigation about why is this and what is going on, and then even more importantly, what can we do about it? We saw over 30% of the kids were African American and Black by race, and 46% were Hispanic children. Next slide, please. This also gave us an ability, if I could have the next slide, please, to use our Children's Research Institute to look even deeper into some of the biology and the science to understand the impact of COVID-19 on children. And I know Dr. Bollard will be presenting some data around that, and other scientists from both Children's National and the NIAID. Looking to the future, I just wanted to take an opportunity, if I could have the next slide, to talk about something that's near and dear to our hearts. So this is the Walter Reed Army Medical Campus that was decommissioned as part of the base closing commission here in Washington, D.C. Many people will remember that building, that big white building, as being the home of the Armed Forces Institute of Pathology. And the United States Army transferred this land and these buildings to Children's National. And we've been building a research and innovation campus devoted to children on this site. It's a one of a kind. As I said, the research and innovation that will be happening there is specifically devoted to children. And we have founding partners that make it very important and tie into the COVID-19 pandemic. That is, one of our founding partners is Johnson & Johnson, and we'll be locating a J-Labs there and be locating 20 to 30 startup companies focused on pediatric research and innovation. As well, J-Labs will be hosting BARDA, part of the HHS grouping, and to focus on vaccine research. And I think many of you know that Johnson & Johnson has one of the earliest vaccine candidates. So we were able, through the good graces of Mayor Bowser, to continue construction as an essential enterprise in Washington, D.C. And this campus will open in the first quarter of 2021, and we'll be hosting researchers from Children's National, from Johnson & Johnson J-Labs, BARDA, the 20 to 30 startup companies I mentioned, the Rare Disease Institute at Children's National, as well as an academic partner in Virginia Tech who will be bringing some of their brain tumor research to this site. Next slide, please. And we think that this is just such an ideal location. The location is at Bear to the north of the Children's National main campus for pediatric research and innovation. And part of that is its proximity to the NIH and the NIAID and all the other institutes, as well as the FDA, all of the other biohealth activities that are happening here in this region. So we're very excited about playing a larger role in having a site, a location, a campus, which will expand on our great partnerships with the NIH and all the terrific scientists and clinicians that are based there, as well as the access to all of the other opportunities here in Washington, D.C., including Children's National and our Research Institute. So with that, I want to thank you for participating today in this ongoing series. We look forward to the rest of the day and to future seminars and meetings that foster such great collaborations and partnerships with our great friends at the NIAID. So thank you with that, and Lisa, I think it's back to you.
Thank you very much, Kurt. And so we're going to save questions for a question and answer session with the panelists at the end of each session. I would like to invite all of the attendees and the other panelists to post their questions in the Q&A box so that that we can be sure to get what's on your mind conveyed to the speakers for their responses. So our next speaker is Dr. Tony Fauci, who probably needs no introduction, but it is my pleasure to introduce him, so I will. Dr. Fauci was appointed the director of NIAID in 1984, where he oversees an extensive portfolio of basic and applied research. He's a longtime chief of the Laboratory of Immunoregulation and has made many seminal contributions to both basic and clinical research on the pathogenesis and treatment of immune-mediated and infectious diseases. He's the author, the co-author, or the editor of more than 1,300 scientific publications, including several textbooks. Dr. Fauci is the recipient of numerous prestigious awards, including the Presidential Medal of Freedom and the National Medal of Science, as well as the Mary Woodard Lasker Award for Public Service. He's a member of the National Academy of Sciences, the National Academy of Medicine, the American Academy of Arts and Sciences, as well as a number of other prestigious national professional societies. Dr. Fauci has advised 6 presidents on HIV, AIDS, and many other domestic and global health issues. And he is, I think, without question, one of the leading voices in the world about the current COVID-19 pandemic. So, Dr. Fauci, I'd like to invite you to make your comments for this symposium. Thank you.
Thank you very much. It's really a pleasure to be here with you today to talk about this very important subject. There are a number of ways to approach this extraordinary historic pandemic. I want to talk a bit today about both the public health and the scientific challenges that we face. So let's get right to it. A few months ago in January of this year, I wrote a viewpoint for the JAMA, and I entitled it Coronavirus Infections More Than Just a Common Cold. And I chose that title not to be facetious, but to point out to the readers that although in January there was an evolving situation with this, at the time, potentially pandemic virus, a coronavirus, but I wanted to put it into perspective. So we know that there are multiple human coronaviruses. Those that are highlighted in yellow are what we call the common cold coronaviruses, namely 4 of these viruses, which historically, over decades and decades, have been responsible for about 15 to 30% of the common colds that each of us repetitively experience, usually during the winter months. And then in 2002 and 2012, respectively, we were confronted with the first 2 of the potential and reality pandemic coronaviruses. The first was SARS, as you might remember, in 2002, from a bat to a civet cat to a human from Guangdong province in China, which then epidemic and then pandemic led to 8,000 cases and almost 800 deaths, spontaneously was able to be controlled by public health measures because its ability to transmit from person to person was not at a high level. Then there was MERS, Middle East Respiratory Syndrome, again, bat to camel to human, still some smoldering cases. But now what we're faced with is the 3rd pandemic coronavirus, emerging again from China in the Wuhan province, very familiar to most people now because of the extraordinary attention paid to it justifiably, because this virus was first identified in January when the Chinese put it on a public database. It's called SARS coronavirus 2 because of the phylogenetic proximity to SARS coronavirus 1. So just to be clear, we all know the disease is called COVID-19 for coronavirus disease 2019. First cases recognized in December, possibly cases even before then, but first officially recognized in December. And as I mentioned, the virus is called SARS coronavirus 2. Fast forward to where we are today. As of yesterday, we now have a truly historic pandemic like nothing we've seen in 102 years since the now infamous pandemic of 1918. There have been over 33 million cases, and now we've reached an extraordinary landmark of 1,000 deaths globally. The United States clearly has been hit harder than any other country in the world, with the last count as of last night, 7 million cases and over 200,000 deaths, with the distribution throughout the United States and its states shown by this heat map shown on this slide. There's some interesting differences between the United States and Europe, which tells us a little bit about the status of where we are right now, which is really important. If you look at the peak of the European Union, which peaked a little bit before us, it went up to the number of cases shown there. But when they shut down their I wouldn't say their country because it was a wide range of countries, their baseline went very, very low to a couple of thousand per day. The United States had our peak, which was dominated early on by the northeastern corridor, particularly the metropolitan area of the New York City, which literally had maybe 40% of all the cases in the country at one time. However, note that when we shut down, if you look at our baseline, it plateaued at around 20,000. Then when we try to, quote, open up the economy, we had the resurgence of cases, including and dominated by southern states of Texas, Florida, Arizona, and California, such that the daily count went up to 70,000 cases a day, has now come back down. But unfortunately, for the last few weeks, literally, it has been stuck at around 40,000 new cases a day and anywhere between 600 and 1,000 deaths per day. Why is that a difference between us and the European Union? When you go back and look at the extent to which we shut down by measuring people's mobility over time, look at the attendance at parks and outdoor spaces comparing the United States in the dark line to Italy and Spain, which are representative prototypically of the European Union. If you look at workplace visits, again, the United States did not shut down nearly as much as our European colleagues. And when you look at things like going to a grocery store or to a pharmacy, again, a major difference between the United States and the European Union countries. That's the epi. Let's talk a little bit now about some of the things we've learned over the weeks and months as it has evolved, something we did not fully appreciate early on, that about 40 to 45% of the cases are in individuals who have no symptoms at all, not pre-symptomatic, but no symptoms at all. The thing that complicates this even more is that when you do modeling, we know that a substantial proportion of spreading and cases in transmission occur from a person without symptoms to someone who is uninfected. So let's take a little bit look at transmission, which we're all too familiar with right now. There are certain things that we need to do, and I could summarize it all in one slide. I call them the 5 fundamental things that should be universally addressed, namely the wearing of masks and cloth face coverings, physical distancing. We talk about social distancing. I think that's the wrong word. We can communicate the way you and I are doing right now, but it's the physical distancing because of droplets that in essence, when you're 6 feet or closer, have a much greater chance of transmission. Avoiding crowds and congregate settings, particularly indoors bars, which have become a clear hotspot for transmission. Anything you do, if possible, outdoors better than indoors, and frequent washing of hands. The clinical manifestations are protean, but very similar to a flu-like syndrome, if you look at what's shown on this slide. However, there are also some interesting symptoms and signs that, in fact, are unusual for other diseases, including the peculiar loss of smell and taste, which usually precedes the onset of respiratory symptoms in a certain proportion of individuals. The other thing that's confounding about this is that you have such a wide range of symptom and manifestation, from nothing at all to mild to severe. About 80% of the symptoms and signs are, in fact, mild to moderate in the clinical course, about 15 to 20% severe to critical, with the fatality varying from a few percent to about 25% in people hospitalized requiring intubation and ventilation. The manifestations of severe COVID-19 are shown here. We're very familiar with them. The dominating one being the acute respiratory distress syndrome. But the more we see and the more clinical experience we get, we see a variety of manifestations, including hyperinflammatory states, cardiac dysfunction, sometimes leading to acute cardiac arrest due to arrhythmias or very progressive cardiomyopathies, neurological disorders, hypercoagulable, either microthrombi in small vessels or acute strokes with thromboembolic phenomenon, kidney disease, and something of obvious interest to the pediatric physician population is the multisystem inflammatory syndrome in children. Over 800 cases now reported. This is a case report from the New England Journal of Medicine from a couple of months ago, about 186 patients, serious disease resembling Kawasaki's. About 80% of those individuals required intensive care, and 20% of them required mechanical ventilation. When you look at the epidemiology of COVID-19 in United States children, it represents about 10% of all reported cases. But as we all know, the hospitalization rate is really very low compared to adults, and the death rate is extremely low. However, there are a lot of things that we do not know about this virus, including the long-term consequences, which we're seeing mainly in adult, but in some pediatric patients. So we really have to keep an open mind as to the long-term seriousness of these infections in children. Now, we have a study at the NIH called the HERO Studies, which is to determine the incidence of infection in children and whether or not underlying conditions like asthma or allergies affect the development one way or the other of infection, and what percentage of children who go on to develop. Because this still, as the literature keeps evolving, is not 100% nailed down about everything we know about the role and the rate of children getting infected and how well they transmit at different ages to adults. The other issue with this disease, COVID-19, that again is perplexing, is something that has been called long haulers, or individuals who recover from various degrees of severity and wind up with symptomatology lasting weeks to sometimes months and maybe even longer because we don't have the full spectrum yet. These individuals complain of things like myalgias, fevers, fatigue, and what's referred to as brain fog, or inability to concentrate. Again, reminiscent somewhat of myalgic encephalitis and chronic fatigue syndrome. The people at increased risk for severe illness are shown here on the slide. First, as I mentioned, with relationship to pediatrics, is older individuals. If you look at the rate of hospitalization per 100,000 population, the differences between younger individuals and elderly individuals is really quite clear and striking. In addition, people of any age with certain underlying medical conditions that are shown here, these, according to the CDC, are those that are strongly associated with an increased risk of severe disease. And that includes serious heart conditions, kidney, obstructive pulmonary disease. I point out obesity because that is one that is really clearly associated with a severe outcome of COVID-19. There are other conditions that may confer an increased risk. And again, they're shown on this slide. Pointing out one or two is diabetes and hypertension, which appears actually to be a very important comorbidity in this regard. And again, we have with COVID-19 things that we've seen with other diseases, a clear-cut racial and ethnic disparity, where African Americans, Latinx, American Indians, Alaskan Natives, and Pacific Islanders suffer disproportionately, particularly when you're talking about a higher rate of infection, but also, again, using the parameter of the rate of hospitalization per 100,000 population. Take a look at Hispanic, Latinx, Black hospitalization compared to white, 359 and 357 compared to 78, a clear-cut disparity there. Moving on quickly to therapeutics, the NIH has put together what's called a treatment guideline panel of getting experts in various elements of this disease, clinical experience, ICU docs, people who understand the disease, to meet regularly to form what's called a living document to put out in real time clinical data as it's published and even expert opinion. This is easily accessible by the link shown on this slide, COVID-19 treatment guidelines.nih.gov. There are 2 drugs that have now been recommended by this guidelines panel. I'll get to them in a moment, but also there are examples of other things that are under clinical trial right now, things we don't have an answer to. And those are direct antivirals, convalescent plasma. Still now, there's an EUA for that, but we still don't know definitively the answer of the safety and efficacy. Probably quite safe, not so sure about the efficacy. Then there's hyperimmune globulin and a lot of promise in monoclonal antibodies, as well as cytokine inhibitors and anticoagulants. I want to spend just a couple of seconds really on 2 of these drugs. And one of them is remdesivir, which was the first randomized placebo-controlled trial of people with advanced disease, namely in the hospital and lung involvement, showing a significant diminution in the time to recovery. Another randomized placebo-controlled trial was done by our colleagues in the UK, showing that dexamethasone, when given to individuals with advanced disease, namely on ventilators or requiring oxygen, that they have a significant positive impact on diminishing the 28-day mortality. Of note, there was no benefit in early disease. In fact, some deleterious effects, which goes along with what we know about the pathogenesis, namely that you need an antiviral effect early on, keeping the immune system intact. And later on in disease, when you have advanced disease, it's the hyperactivity of the immune and inflammatory response that needs to be muted. And then finally, vaccines. We've taken in the United States a strategic approach to COVID vaccine research and development. There are 11 candidates throughout the world that are in phase 3 trial. There are 5 of these, of the 6 that the federal government is supporting one way or another that are in phase 3 trial. They're shown here on this slide. 3 separate platforms: nucleic acid, represented by mRNA, viral vectors, such as chimp adeno, human adeno, VSV, and measles, and the typical protein subunits with and without an adjuvant. Right now, 2 of those trials, the Moderna and Pfizer, went into phase 3 on July 27th. So we would anticipate that we would have an answer as to whether or not we have a safe and effective vaccine, likely sometime in November or December. We could hear earlier, maybe October. I don't think that will happen, but it is certainly possible. It is more likely that it'll be later on in November and December. Since vaccines have already been produced, it is likely that there will be doses available towards the end of the year and the beginning of the year. Again, if we have a safe and effective vaccine, because obviously we need to approve it. I'm cautiously optimistic that we will, based on encouraging data from the phase 1 trials showing the induction of robust levels of neutralizing antibodies. Let me close with this last slide, in which an individual can access things that are going on in the prevention network. And if anyone has any interest in volunteering for any of the prevention trials, you can indicate your intention to do this. There's no obligation. You could just sign up to get more information about volunteering for these trials. So I'll stop there. And again, thank you for giving me the opportunity to make this presentation.
Thank you very much, Dr. Fauci. Our final welcoming comment is from Dr. Chris Austin, who is the director of the National Center for Advancing Translational Sciences, or NCATS, at the National Institutes of Health. Dr. Austin leads the center's work to improve the translation of observations in the laboratory, the clinic, and the community into interventions that reach and benefit patients. Dr. Austin's career has spanned the spectrum of clinical research in both the public and the private sectors. He's trained as a neurologist and a geneticist. He's worked with the pharmaceutical company Merck. And in 2002, he joined the NIH as a senior advisor to the director for translational research at the National Human Genome Research Institute. While at NHGRI, Dr. Austin founded and directed the NIH Chemical Genomics Center, the Therapeutics for Rare and Neglected Disease program, and the NIH Center for Translational Therapeutics. NCATS was launched as the successor to NCRR in 2011. And in 2012, Dr. Austin became the inaugural director. Dr. Austin has many accomplishments and awards. He is a member of the National Academy of Medicine, formerly the Institute of Medicine. And it's my pleasure to introduce Dr. Austin to you to give his overview and welcoming comments. Chris?
Thank you, Lisa. It's a real pleasure to be here. And I really like how this introductory session has been done, because the back-to-back sessions with Tony and me really reflect the partnership of NIAID and Children's National. I have the privilege, among other things, running NCATS to run the CTSA program, of which the CTSI there at Children's is a prominent member, and Lisa being a prominent investigator among this on this network. So what I'm going to tell you about today is some of the work that we've done, a lot of which is in collaboration with our colleagues at NIAID, trying to take a translational approach to this. And what I mean by that is that, as Tony pointed out, this pandemic, like the opioid epidemic before it and ongoing, has revealed a lot of quite general issues that we have in the process of going from a fundamental discovery to an intervention to getting it all to all the people who need it. And so NCATS has taken its traditional collaborative approach to this pandemic, and I want to give you some idea of what we've been doing in this area. So on our website, I'm not going to go through this, but we have a listing if you're interested in how we've approached this problem. Essentially, the way to think about it is that NCATS is an engine that has all kinds of innovative technologies and approaches and paradigms to improve the efficiency and effectiveness of the translational process, all the way from target validation to public health. And when COVID came out, we pivoted many of these really disease agnostic capacities to this really unprecedented in our era pandemic, like that Tony described. And I'm going to tell you about a few of these in the next few minutes. So I'm going to start with the preclinical side, and I'm going to end up on the clinical side. On the clinical side, the issue here was that a lot of people were and are doing drug screening, compound screening against a variety of intact virus assays or surrogates down to molecular targets from the virus and also from the host. And the issue here was the typical one, that those many labs were all running compound screens simultaneously, but the results were not shared quickly or completely, including the negative data, which is critical, and they're not easily available to the scientific community. And this, of course, limits their impact and delays how quickly we can bring treatments to these patients. And we thought, I think, like all of us on the phone, this is just not acceptable in this current era. I don't think it's ever acceptable, personally, but particularly in this era, we cannot be satisfied with the let's wait a year and publish a paper before these data come out. So what we did at NCATS on the left side is to develop both in our labs and collaboratively assays for many of the targets in the SARS-CoV-2 genome and ran those across the NCATS pharmaceutical collection, which is a collection of every drug ever approved for human use worldwide, and then made those data immediately available to the world and in a form which you can see there on the right side. And I just encourage you to go to the website. You can see great detail on each of the assays, including the protocols that we use in the lab, not the sort of Cliff Notes version that one finds in a paper, but all the details, all the secret sauce that you need, and all of the positive and negative data on all of these compounds, many thousands of compounds. And then one can sort them by drug type or by target or mechanism of action or whatever you like. And that's all available. Secondly, this was something that we started right before COVID-19 came out, but it couldn't have possibly happened at a better time. So this is something that we've been developing with our colleagues in the infectious disease division at the FDA for the last 3 years. That is a mobile app that allows healthcare workers, initially focused on neglected tropical diseases was the initial intent, that allows healthcare workers to share their clinical experiences using approved drugs to treat diseases other than the ones for which they were regulatorily indicated. That is what the label says. And this is an app called Cure ID. And I would urge you to go to that website down there or go to the App Store or get it on your Android. It's quite a cool app, if I do say so myself, that allows case reports, allows discussion among principally infectious disease docs and immunologists and rheumatologists across the country, data on clinical trials, et cetera. And this happened to be launched at a joint NCATS FDA meeting the first week in December last year. And as you heard Tony say, this is about contemporaneous, we think, with the first COVID cases a few weeks before the first COVID case was reported. And of course, so then we added a module for COVID-19 along with all the other illnesses and infectious organisms you can see there. And particularly, I would say, in the early days of the pandemic, when there were drugs being used in a variety of settings, community hospital settings all over the world, in addition to academic settings, with no systematic data, because it simply hadn't been that long. this app was particularly important. Now that we have more systematic data of the sort that we're all aware of and Tony was talking about, I think the importance has gone down a bit. But you'll see a lot of our favorites here, leponavir, ritonavir, our old favorite hydroxychloroquine, azithromycin, a lot of the old favorites you'll see there. And back in June, the Critical Path Institute launched an expansion of this drug repurposing collaboratory to focus on not only COVID-19 but other infectious diseases. So stay tuned, and I hope you'll download the app. It's a very cool thing, and I get an update on this every morning, and it's always fascinating what I learn. Switching to a survey which we've been doing with Matt Mamoli and his group and colleagues at NIAID and colleagues at NIBIB and at Frederick, a very large sero survey to evaluate the proportion of the healthy population that's been exposed to SARS-CoV-2 by measuring antibodies. And I think you can imagine what we're interested to learn here, learning the extent of COVID-19 in people who have not been diagnosed. As you heard Tony say, at least half of people, perhaps more, who are infected are not diagnosed with COVID-19. Certainly don't have a positive test. And how these distribute geographically, demographically. You heard Kurt Newman talk about their experience in the drive-through line there at Children's and the health disparities which have been experienced. And of course, assisting investigators to understand the immunity and correlates of protection. So where are we here? This was really a success story for clinical research. When Matt Mamoli first put this out, he was hoping for perhaps 300 to 500 people who would volunteer. Within several weeks of having this opportunity available, 400,000 people had volunteered. Which created a nice problem. I wish I had that problem with every clinical trial. Recruitment is usually not quite so simple, but it really was in this case. So then we had a real problem. How do we get a representative sample or as close to it as possible? So we brought in the CTSA investigators who know a lot about this at Birmingham, University of Alabama at Birmingham and at Pittsburgh. And working with them, we got a proper sampling of the U.S. population. There's a highly specific testing method, which I don't have time to go into, but we have been quite neurotic about validating this testing method, and all those data are available too. It's all been published. We decided to go with 10,000 people. All of those 10,000 people have been recruited. We've received and run 8,500 samples. The other 1,500 samples are all in various stages of getting submitted. It really illustrates the logistical issues here. It's not a scientific issue; it's a logistical issue of getting these samples sent to us and where they can be assayed. And again, like I mentioned with CPATH, having picked up expanding the Cure ID app, the NCI through their serological Sciences Network has funded an ongoing study of this to look at these same people at 6 months, 12 months, or perhaps ongoingly. Really depends on how this pandemic evolves. Our tissue chip program, which you may be aware of, microphysiological systems program, pivoted as well. Don Ingber and his team at the Wyss Institute had previously been funded to develop a human on a chip, which is quite well developed now. And its response to influenza. And when COVID-19 came out, we funded them to pivot to SARS-CoV-2 and to test repurposed drugs. And you see a number of drugs that inhibited viral infection under these kinds of physiological conditions on the chip, and a number of drugs, including, interestingly, hydroxychloroquine, that did not work in the system consistent with the clinical data. And all this has been published there as well, as you can see the bioRxiv paper there in the bottom right. Proof of concept, at least, that tissue chips could help identify existing drugs, both for now and the future. Tony mentioned the issue of convalescent plasma. I think you're all aware, hope you're all aware of the issues here, and the important thing is we just do not know about whether convalescent plasma We do not know whether convalescent plasma is efficacious in treating COVID-19 disease or not, and we don't know particularly what populations it might be useful for. The observational data are interesting. Certainly safety does not seem to be an issue, but efficacy certainly is. So NCATS started back in April funding a trial at NYU and Einstein in the peak of the NYC caseload with a target enrollment of 300 at NYU and Einstein, and they got to about 180, and the public health measures that were put in place there were very effective and cratered the enrollment. So we were not able to continue enrollment. So through Operation Warp Speed, thank you Operation Warp Speed, we were able to expand this trial to additional sites in Texas and Florida, and we're considering additional sites now that the pandemic is continuing its march through other states in the country. And you can see the current enrollment there. Vanderbilt started its own trial with a catchy name there. Also started in April, had the same dynamic, decrease in caseload in Nashville, limited their recruitment. So through all of this is being done through the CTSA program and the Trial Innovation Network, both of these trials. That's the only reason we can do this is that the CTSA network was ready, and the Trial Innovation Network, which connects all of these hubs together, these 60 organizations across the country, each of which, like Children's, has many spokes into the community and community hospitals and public health departments. We were able to capitalize on this capacity, on this really unique national resource to expand this trial. Again, with Operation Warp Speed funds, and we're currently bringing on up to 50 sites to fully enroll this trial, which would be about 1,000 people. The last trial I just want to tell you about something that I don't think Tony mentioned explicitly, the active program accelerating COVID-19 therapeutic interventions and vaccines. This is a FNIH, NIH, pharma collaboration. There are a number of trials, 5 of them being run through this, 3 of them through AID, 1 through NHLBI, and 1 through NCATS. NCATS has the Active 1 trial. It's a master protocol trial of multiple immunomodulators, and you can see the candidates there at the bottom left and the primary endpoints there. This is about ready to open, we hope, this week, or at the latest beginning of next week. So please stay tuned for when that happens. Again, the only reason this was possible is because organizations like Lisa Guay-Woodford's are present and ready to go. We're focusing on adults here, so the Children's is not currently involved in these trials, but the point being that only through CTSI organizations throughout the country that are already set and ready to go were we able to get this trial going. I want to finish with an informatics effort, which also illustrates the remarkable capacity that the CTSA program has for advancing translational goals in public health. So I think we all would have loved, in the early stages of the pandemic, to have close to real-time electronic health record data from hospitals around the country to tell us what kinds of populations were being affected, what kinds of therapies were being used and might be effective or ineffective in patients who were being treated with COVID-19. And we didn't have those data because there was no network to gather the data. So we have taken advantage of if you will, of this opportunity and this imperative that I think all of us feel to get over some of our hesitations to share data with each other, aided by the public health exemption, to put this national COVID-19 cohort collaborative together. So what is this? This is a collaborative which currently involves over 500 people at hundreds of institutions across the world, across the country, focused within the CTSA network, but involving other partners as well now. to make vast amounts, and I'll tell you what I mean by vast in a minute. It's intentional word, of clinical EHR data to understand and improve patient care for COVID-19. Without getting into the gory technical details, it's a different model from the the federated models that you may know about, like PCORI or ACT or OMOP. This is a centralized secure data enclave that provides access to medical record data from people diagnosed with COVID-19 or tested for COVID-19. And it allows you to do all kinds of things that you can't do in a federated platform, including machine learning and AI and recursive testing and more open-ended questions of the sorts that physicians and clinicians really want to ask, instead of a Google is A related to B or yes/no questions that a federated system is very good at. And again, it harnesses the CTSA hubs and an informatics part of the CTSA program called the Center for Data Health, or CD2H. So what's this different? This is actually from the dashboard yesterday. You'll see the number of patients in this now, 600,000 patients, over 62,000 positive COVID patients. That's from only 14 of the 62 sites that have signed up to deposit their data. So you can do the math there. We expect in the end, there will be about 5 times as much data in this enclave. The limitation now is ingestion, data cleaning, the kinds of things that those of us who work with databases are well familiar with. And I know you probably can't see this, but if you look at the slides afterwards, you could see a really wonderful breakdown by gender, by age, by race, ethnicity, et cetera. And so it really is robust in scope and scale. It's all harmonized. Importantly, we take data from no matter what the data platform, the data model that the individual institution uses. We have built a harmonizer. So if you use PCORI, fine. If you use OMAP, fine. If you use ACT, fine. Whatever, Trinetics, whatever you use, we will do the work to make this harmonized into one OMOP platform that allows us to analyze all this at once. And then we have a collaborative analytic platform within the enclave to allow the kind of team-based research and machine learning that you need to do to really take advantage of this kind of resource. And importantly, and again, I don't have time to get into this, but we have realized from the beginning that if this platform is not rock solid, secure, and we protect patient privacy, we will have failed, not only for this program, but for future ones. So we have applied the highest levels of security for people to get data into and out of this program. And you can see more details there at the link. I mentioned this before. This is how it works in 4 steps. We do a data transfer agreement with the organization. You don't have to change your data into anything. We do all that work for you. Data acquisition, transformation into an OMOP platform. And then we have within this enclave a data use agreement that individuals who want to access the data use and go through a data access committee in order to get access to the data. We're also creating a synthetic data set, which I think will be very exciting, which will allow us to release these data without getting particular restrictions, because these don't reflect real people, they're synthetic people in this case. So is the N3C open for research? Yes, it is, I'm glad to say. It opened for research on September 3rd. We thought about this a little bit because, as you saw there, of the 63 sites there you see on the right who are onboarding data, we only have 14 now. But we thought, well, gosh, 601,000 patients with 62,000 COVID patients, that's a lot of data. So you may want to, depending on the question that you want, you may want to ask the question now. You may want to wait a couple of weeks because it goes up by several 100,000 every week. Or you may want to ask the question ongoingly. Every few weeks you can ask the same question. More than 63 institutions, as I mentioned, have agreed to transfer their data. In addition to most of the CTSA hubs now, that also includes a number of the hubs from the IDEA States Clinical Translational Research Program, which is has a great deal of disproportionately affected patient populations in the Midwest and the South, ones that the CTSA program doesn't have access to. So it's a really wonderful complement. And you can see there about how to apply. So that's what I'm going to tell you about today. It really complements, I think, what Tony was talking about. In all of these things, as NCATS always does, we collaborate quite closely with our domain experts in this case being infectious disease and immunology. And I want to thank Tony for all of his collaboration, Cliff Lane, who I have gotten to know quite well. What a pleasure that is through this process, and others at AID. So I'll stop there.
Thank you very much, Chris. I do want to thank all of the speakers for a very comprehensive overview. We have the perspective of a major children's hospital about partnerships with the NIH that I think is going to be increasingly an extramural, intramural paradigm. NIAID about the clinical science and the basic research around SARS-CoV-2 and the infectious disease that it causes, and NCATS about translational platforms. And I think these comments have elegantly set the stage for the rest of the day's presentations. I would say that we're running a little long, and so I think maybe I will synthesize some of the questions in the question and answer box and pose a question for Dr. Fauci and for Dr. Austin. So first to Dr. Fauci, let me start with a health equity question. Dr. Fauci, the observed ethnic disparities. When adjusted for comorbid conditions, do hospitalization rates among ethnic groups compare? In other words, is this something about their genetics or their environment of these populations, or is it about the health care that they receive and that they have a higher burden of comorbid conditions?
You know, it's the latter. It's the high burden of comorbid. There are two things. I refer to it really as a double whammy. Minority populations, both African American and Latinx, actually have a higher incidence of infection to begin with. So their numbers start off larger. And the reasons for that are almost certainly that you don't like to generalize, but in this situation, it's appropriate to generalize, that the jobs that they generally have put them on the front line of being out there exposed to people on a person-to-person basis, less likely to be able to pull back and do work at home virtually with a computer. So their exposure is more at risk. Once they get infected, if you look at the rate, the incidence, and the prevalence of the comorbidities in the minority population, it is significantly greater than in the population in general. And I refer to several among many. One is obesity, the other is diabetes, the other is hypertension, chronic lung disease, cardiovascular disease, and chronic kidney disease. All of those, if you look at the rate, incidence, and prevalence, it's greater. So they're getting hurt with disparities from two ends. One, a greater rate of infection, and two, a clear greater rate of comorbidities, which makes for that very disturbing slide I showed of literally 300 and something per 100,000 compared to 79 in whites, a very, very large disparity.
Thank you very much. That's an incredibly important answer in this time where we see such disparities across the different racial and ethnic groups in the United States. Chris, I'd like to ask you a question. You so elegantly described the various translational platforms, N3C, CureID, that NCATS has stood up quickly using the power of the CTSAs. What it seems to me, though, is that there's another sort of benefit of the response to SARS-CoV-2 and the COVID-19 pandemic. And that is the kinds of partnerships that NCATS, serving as the sort of the translational nidus, the translational organizer, is orchestrating across the NIH. And so you've talked very beautifully and elegantly about the partnerships with NIAID. But you mentioned that this is a systemic disease. And so I'm wondering if you could briefly touch on the partnerships in terms of clinical trials to address the kidney-related issues in AKI, of course I started with that, the cardiomyopathy and the coagulopathy with NHLBI, the brain fog with NINDS, and it goes on and on and on. Is this kind of serving as a tipping point for now really integrated, fully integrated NIH response.
Yeah. Well, thank you, Lisa. You just gave my speech.
Good.
Yeah, it really is true. It really is true that you know, NCATS has the privilege, if you will, of not having a specific disease to cure. You know, Tony has diseases that affect people to cure. Our disease is the system. Our disease is the inefficiencies and ineffectiveness in the entire biomedical research translational ecosystem that we have. So we are able to build these and pay attention to and build solutions to these kinds of problems which cut across every disease. And one of the wonderful things truly about COVID, I always hesitate to use that word, but I think you all know what I mean. A true silver lining of this pandemic, in my view, is that these kinds of partnerships have happened in a way that they have not happened before. And I do hope and believe that this is a tipping point for the CTSA program and NCATS in general to work with other institutes on all kinds of other issues. We've done that in other circumstances, and that's, as you know, Lisa, that's our MO. But I think this illustrates very graphically why this is such a good idea and why a 1 + 1 really equals 3.
Thank you very much. Before I ask Kurt to make some sort of closing comments to this welcoming session, I would like to make 3 points. First, there are numbers of questions that are very specific, prompted by the elegant presentations that we've just heard. And I think that they may be more pertinent for the sessions that will follow during the course of the day. So in the interest of time, I will defer them. The second is that the recordings of the presentations this morning will be available both on the Children's National website and of course our CTSI-CN website. And the third point is that I think it would be very nice, we started with our CEO to bring this full circle and ask our CEO to close this session before we take a 10-minute break. Kurt?
Well, thank you, Lisa, and I really want to thank Dr. Fauci, Dr. Lane, and the leadership at the NIAID for having the foresight to create this partnership with Children's National. We didn't know there was going to be a pandemic and that the pandemic was going to have such an impact on children. And to see their leadership, hear the presentation today by Dr. Fauci, and looking forward to the rest of the presentations is so important to our children, our children's health. And, you know, it's just a beautiful example of collaboration and partnership. And then all the great work that Dr. Austin's doing with all the CTSIs and NCATS, and Lisa, your leadership with our own pediatric-focused CTSI, you can really feel how the beauty of science and investigation. And there's so many unanswered questions, but I'm just confident that as we hear the presentation today, we're on the right pathway to make terrific progress. And in our case, for children and families. And it was pointed out there's so much we don't know, and then there's the hidden impact on mental health and all of the other issues that are relevant to a pandemic like this. And so being aligned with the NIH and the NIAID and NCATS, it's so important for Children's National. We look forward to that continuing partnership. And again, many thanks, Dr. Fauci, for your leadership in creating this partnership.
Thanks very much, Kurt. So that will close our welcoming session. We'll take about a 5-minute break and we'll resume at 10:10 with the next session, immunobiology and host genomics, that will be moderated by Kath Bowden. So again, thank you very much, Dr. Newman, Dr. Fauci, and Dr. Austin. This is a great kickoff for a great day.
Thank you.
Thank you very much. Good to be with you. Thank you for having us.